Well, that's, in the case you didn't know, Emerson, Lake and Palmer, prog rock from the 70s. I was brought up with it. In fact, the IT guy here did a concert in this room not so long ago. Was it last year? Something like that?
About 7 months ago.
7 months ago, where Greg Lake himself was in this room playing to a birthday party. So we're in hallowed halls. Thank you very much for the invitation. It's always a pleasure to come to the meeting. And what I'm going to do today is walk you through the thinking that came to 2 clinical trials. I sat in front of what was first of all a scientific talk, trying to sort of simplify it, and actually I couldn't. So what I'm going to do is show you what I'd show the scientists, and then I'll try and explain to you a little bit about it. Do interrupt me if you don't understand as we go through. So I'm based in 2 centres, really, in a basic science laboratory called the Dunn School of Pathology, which is where penicillin was first purified and where cephalosporin antibiotics were first discovered. And when penicillin was purified by Florey, the purified material was taken from this laboratory and walked across to the Radcliffe Infirmary, an old hospital in the centre of Oxford, and 2 patients were then just given the drug. There was no consent, and the drug worked temporarily, and there was some benefit, but we didn't have to go through all the rigmarole of the clinical trial directives to get penicillin started. In fact, if we did do penicillin in a clinical trial, there'll probably be major concerns with its toxicity, along with drugs like aspirin. So the history of where we've got to now is really about protection of patients so we don't experiment on individuals inappropriately. And that necessarily is a legal perspective, and that necessarily leads to a very prolonged process of getting trial approval. And I'll tell you about how long it took to get these 2 trials done. So this little university in the center of this great country is now a biomedical research center of a world-class nature. And it reflects actually the case in many centers in the UK. We are disproportionately able partly because of the NHS, partly because of the culture, but also because of the underlying great scientists that are around. Some of those initiatives have been very successful, but many have failed. But there's still a great energy to bringing new bright minds into new problems. And for many years, Oxford doesn't really participate in any sarcoma or bone cancer research, really, apart from one individual who's been working on what are called giant cell tumours of bone. But about 5 or 6 years ago, a number of us got involved, and this is, in a sense, a reflection of the story of where we've got to. The whole piece in Oxford changed because it suddenly recognized that to be a competitive environment, it had to work on an international scale, and it had to seek huge amounts of investment. And really, over this last decade, there's been a shift from the center of the town, which is the university part, the old part, to the Headington campus, where a large number of hospitals have been and research centers have been built, such that now, depending on what metrics you use, the most successful biomedical research arena in the world is actually this one. And that's happened over a generation. It's taking a huge amount of investment and commitment. And so the job of people like me is to be in the clinic, but also to make sure that the knowledge and the abilities of people in this environment are brought to this problem. And so the traditional separation of how academic or research environments work out is that there's been essentially 3 silos. Those in biomedical sciences, basic research scientists that can't have a conversation with you and you can never get eye contact, to medicine in developing pharmaceutical companies where men in suits want to take money from you, and really a dedicated population who really want to improve patient care and are at the front line and really understand it. And really these things were not connected. And so over this last time in Oxford, it's very clear that we needed to invest in large-scale clinical trials. And they're very important because that's what changes practice. And if you go across the country, we're actually quite enriched by expertise in that area, some of which are connected to the Bone Cancer Research Trust, people like Jeremy Whelan or Mike Leahy or Ian Judson. The other area that we needed to embark on was really this area of experimental medicine. How do you give the right drug to the right patient at the right time? Because not everybody can have the same drug, not everybody is going to have benefit from it. And so a shift in the thinking has been to this experimental medicine, and this is where the new bill falls in as to how to shape that. How do you regulate that? Because in the end, it's a risk. You're taking a risk of trying something out in the hope that it's going to be beneficial. And then the other area that we really recognized needed a lot of investment was this connection. How do you translate the basic sciences with the men in suits to actually improve patient care? And this is about actually identifying targets that are relevant for the disease you're interested in. And that's an area often where the charities are very good at investing in really these three areas. So if I were to sort of reflect on what was said earlier, that the UK government has really tried to support the pharmaceutical industry. It's supported basic science, particularly the Wellcome Trust and the MRC, and we have a lot of investment in frontline patient care, and there's a lot of research related to that, which is very relevant. But these three areas are where the step changes happened in the UK. And maybe because the UK is so successful is because of this connection from what are the real clinical needs and how can we use our resources to apply to that. So that's a bit of a heads up on where the thinking was. And so this is, everything else is derivative from that. So the way Oxford set itself up was that no hospital was to be set up without research embedded within it. No hospital setup was to be established without a linked clinical research arena. And so this is the case for the service in cancer and the cancer research laboratories, in pediatrics, young adults, neurosciences, surgery, musculoskeletal surgery as well. So they've all got a link. And so this becomes a matrix, a network of interrelated expertise. And at any one point, if you're trying to solve a problem, you don't know who's going to help you the best. But if you have all that available, it really fast tracks your ability to translate. So I'm going to talk about the two main tumors, Ewingsarcoma and Osteosarcoma. And this is where I'm going to show very straightforward scientific slides. And they look very complicated, but they're actually very simple. So for Ewingsarcoma, the data that shows the curves there is quite old. But it hasn't changed a great deal. It's probably slightly better. But on the bottom is time in years, and on the y-axis, that's the vertical axis, it goes from 0 to 1, 1 meaning 100%. So if you take 100 patients with Ewingsarcoma, you can split them into groups, those that have no metastases and those that do, and particularly of those who have lung metastases. If you look at the top line, that is each individual patient, each person, and how long they survive after their diagnosis. And as you can see, it sort of flat lines around about 50%. We're a bit better than that now. We're closer to 65%. And that's mainly because of standardization of chemotherapy, the introduction of large-scale clinical trials. But what's clear is that if you're unfortunate enough to have a metastasis, a spread of the tumor, then things look worse. For a period, you may be okay, but eventually it flatlines, at about 20–25%, which actually is quite interesting, that you can have a spread of a cancer and still be fine, 15–20 years later. So it's not all negative, it's just we've got a lot of hard work to do. And the question is, how do you turn that survival curve at the bottom up to one which is closer to those that don't have spread of the tumour? Now, the thing that's changed our whole understanding of everything is DNA, the code for us as individuals, and the mistakes that happen in the DNA, that in each little cell of our bodies, that then change the behavior of that cell and make it into a tumour. And it makes a tumour because the cells divide and lots of cells join together to form a lump. And the driver mutation, the mistake, we know a lot about. It turns out to be a break in the DNA in two sites that then join aberrantly. So they shouldn't really be together, but they become together. And that's called a chimeric transcription factor. It's just a word. But we know where the breaks are, and it's between two main genes called EWS and FLI1. And if you're diagnosed with Ewingsarcoma, the guys in the laboratory do a special test called an in situ hybridization test that tells you whether that mistake is present or not, and that tells you then whether that's Ewingsarcoma. So even if you have this mistake, it doesn't mean to say that your Ewingsarcoma is going to behave like everybody else's. So there must be other things going on in the tumour. We know that the different types of these translocations have very little impact on your overall survival. And this is comparing a number of these here in a project that actually involved a large number of the UK team and a big European trial called Ewings. And so and you can see here the survival's a bit better because this is more recent data published in 2010. You can see that really the lines are pretty close together, depending on the different types of mistakes.
Sorry,
yeah.
Can you go back to the DNA slide?
Yeah.
Does that break in the DNA only happen once the sarcoma's there?
Yeah.
Or is it there before the diagnosis? Presumably it's there before the diagnosis. And what I'm really asking for is can it be screened?
Yeah. So it's not, okay, so there's two aspects to that. As individuals, we all have a slightly different predisposition to Ewing's sarcoma, although Ewing's is very rare. It's rarer in populations such as Africans, okay? And we've discovered, in fact, a Frenchman discovered, the genes in us all that may be increasing our risk of developing Ewing's sarcoma. Now, when Ewing's sarcoma happens, it doesn't happen in the whole body, it just happens in one cell. And this aberrant cell then develops this mistake, reprograms the cell, makes the cell divide. So it's, once the Ewing's sarcoma is formed, that's where the mistake is, and really nowhere else, okay? So does that help answer the question? Yeah, okay, cool, all right. So what do you then do to try and block this mistake? It's unique. It separates the Ewing sarcoma cells from the normal cells. So if we focus on that area, then we've got a treatment that should only hit the Ewing sarcoma cells and not the other normal cells in your body. And we can either go for the breakpoint, the EWS-FLI1 chimeric gene. We can go to the effect of that gene on other genes, or we can go down to even further to the pathways and the signals that the cell makes as a consequence of the EWS-FLI1. And then we can even go for things that are completely irrelevant, but we don't fully understand at this moment. And so what we've decided to do initially was to start to formulate a little thinking around the signaling pathway, because there's a lot of drugs that are in development that can be dependent on EWS-FLI1, and if we target that, they would be separate from the normal cells of the body. And this is a complex pathway. You don't have to worry about it too much, but I'll go straight to this one. So indeed, this is what happened a little while back in what's called an early phase trial of an antibody, which is a protein that's given as an infusion, targeting one signaling pathway called the IGF-1 receptor pathway. And that is a pathway where there are keys or signals on the surface of cells that you can block. And so a lot of patients with sarcoma, many of which were at the Royal Marsden Hospital, had this antibody, had very little side effects from it, and a number of patients responded. And this is what's called a sort of waterfall plot, if you like. So what you have are the columns that are going up are actually patients that have had the disease, that had the treatment, but the disease has progressed. And on the other end are folk that have had the disease and the treatment, and they've responded. And the length of the bar is how well they've responded and how long they've responded for. So it was clear in this mixed group of sarcomas that there were people responding really well to this one antibody, this non-toxic treatment. It's not like chemotherapy. And some of which are still on this treatment today, many years afterwards. So, and in that group were really only the Ewingsarcoma patients. So this was declared as a miracle, because here we had a non-toxic treatment that in a very small subset of patients could be treated every month, that they could be control of the disease without any additional treatments. And this was in the context of, finishing lots of treatment or relapsed disease after many years of treatment. So this was not an easy population. This is a serious situation. So to get just an antibody to work like that is amazing. Don't worry about the graphs on the other side. But after that came a number of other companies and trials. Don't worry about that. This is an example of, so another company, Roche, the previous one was Pfizer, had another antibody that did the same trial, and they could identify a few patients where there was a remarkable response. You can just barely see the shadows on the CT scan of the chest, and you can see the shrinkage of the tumor just with the antibody. But if you look at the survival curves, as I've shown you before, it didn't look very good because many patients were progressing quite quickly. And it was still true then that there was just this very small group, so a rare tumor, and then of that group, maybe 5% at the most getting benefit from this antibody. And that wasn't enough for Roche and Pfizer to say, okay, we're going to invest more money as a commercial company in Ewing-sarcoma. And in fact, what they did was to shut the programs down. So in the community, we had the situation where we were dealing with rare cancer, where we don't get very many opportunities and hits. We've got a clue that there may be something going on here, but we didn't have support from the men in suits. So what do you do as a result of that? Well, there is only one route you can go to get enough money to do more research, and that is to go to European funding. And the reason for that is because with a rare cancer, if you can lobby within Europe, there is a vehicle for collective investment that's beyond each nation state to get enough critical mass of money in order to take research forward. And we've really embraced that opportunity in Oxford. So as a result of this sort of data and the community of sarcoma oncologists and researchers in Europe, we wrote a grant to the European Union, European Commission called Eurosarc, and they gave us 6 million euros. And 6 million euros to do essentially 6 trials. So that's 1 million euro for a trial portfolio. And this was covering all rare sarcomas, so not just Ewing-sarcoma. There was another drug that we knew about that had just been taken over by, I'm telling you all this because, this is how it is in reality. A drug was taken over by a Japanese company, and they were new to the field, And so we went and smoothed them quite quickly before they could work out what was going on. And they committed, so they committed free drug for this drug, which is called OSI 906. It's now got another name. It's called lincitinib. It's a tablet. So it's not an antibody, it's a tablet. It does have side effects. But so a lot of work was done, and this is just some of the data that comes out of the experiments that people do. And what was found that if you take Ewing cells in culture and you put this drug on it, it blocked the Ewing cells in culture. So this gave us some encouragement that we could go back to this question in Ewingsarcoma and see if we could work out what was going on. And so we went back to the lab at the same time and said, okay, look, you know that there's this translocation, this EWS Fly1, but there must be something else going on that changes your sensitivity to the drug. And these are called Circus plots. You've seen them here. They're very common now in the scientific literature. This is how you display DNA and genomic information. And essentially all around the top, on the periphery circle, are all your chromosomes. And the inner line that's a bit wavy are the defects in those chromosomes. And the lines you can see that are joining a crisscross are these aberrant breakpoints or connections called translocations that EWS Fly1 is one of. So in the cell lines in the culture, you can see the translocations, but you can see other things going on as well. And so in the experiments in the laboratory, we've identified candidate mechanisms that confer this extra sensitivity to blockage of this IGF-1 receptor. And we did this by whole genome sequencing of material. That takes about 4 or 5 days to do that. Nowadays, it was costing £10,000 a shot. Now it costs £1,000 a shot. So this new technology is radically changing everything. And after the funding, we set up a trial. And this is the trial. It's called LINES. We wrote the grant in 2010. It was approved at the end of 2011. And we opened LINES about 6 weeks ago. And that intervening period between getting the money and opening the trial was all to do with ethics, legal, contracts, setups, NHS, companies, MREC, and it goes on, MHRA, it goes on and on and on and on. And even though there have been attempts to fast track that, in reality, it's really impossible to fast track it. And so every initiative you do, there's another layer of administration and bureaucracy. And I think it's really frustrating because for people like me, you know, I've got a working life now, 15 years now, right? So if I've only got 3 trials left. I don't think I've got the energy. And it's hopeless. And I think we need to feed back that it's absolutely hopeless. There has to be a better way than this. So this trial, so let me be absolutely clear. This trial is not going to cure Ewingsarcoma. This trial is to find out if we can understand it a little bit better. And so there's 2 aspects to this trial. One is to try and work out what the lincitinib drug is and to pick up where the companies stopped. And the second part is to collect so much information at an unprecedented scale so that the patients that enter these trials, we try and find out absolutely we throw the whole kitchen sink at this to really try and understand what's going on inside the tumors. And that will inform next trials, maybe completely different drugs or strategies. But if we don't mine this material that we get from the trials, then we're missing a major opportunity. So the trial then is an exploratory trial, and the endpoints, that's why we're doing it, are really to determine whether this drug does anything in Ewingsarcoma by biopsying before and after exposure, and by doing PET scans before and after exposure, and to assess the safety and tolerability. it is not a large-scale clinical trial that's going to get it into the clinic so we can treat everybody in the clinic. It's a trial that leads us to that. And it's very intensive. Now, because of the limitations of budget, I mean, this trial and the other trial I talk about have basically eaten up the 1 million budget within about 2 years. And any other additional funding we've got has been from the National Institutes of Health Research, NIHR, in the UK. And we've had local support in Oxford through the BRC and the BRU to do this trial. We've also had local families raise money because we couldn't afford to open the trial in several centres in the UK. And the families have raised £45,000 so that we can put families up and patients in local hotels and allow them access to the trial so that there's no financial penalty to come and get access if they wish to. And that's now happened. So we've had a couple of patients from Liverpool, a patient from Manchester, a patient from Bristol, and we've now got because we're the first of the 5 centres open in Europe, we've actually got a patient from Germany coming next week. So it's anybody with Ewingsarcoma. So there's a very complicated design to this trial, very sophisticated design using working with a very excellent statistician in Birmingham called Cindy Billingham. It's called a Bayesian design. I don't need to tell you too much. And this is the sort of thing we'll be doing. So PET scans, very detailed proteomic analysis, DNA analysis. These are the Circus plots again. So very Bayesian biomarker exploratory. Yeah. Highly detailed, difficult planning. We've got lots of standard operating procedures. This is beyond a normal clinical trial. And so we've put a huge amount of investment into the pipeline of how we'll analyze the data. If we do not turn this one opportunity into 3 or 4 different clinical trials with new opportunities for treatment, then we will have failed. And so that's why we've set it up that way. Very detailed. We've had to invent new technologies to try and analyze the data on very small biopsies. Actually, it's been quite innovative. It's got a lot of people involved, bioengineers and so on. So we're going to do the multiple cell lines at the top and the biopsies in the clinical trial so that if we do the same analysis for both, anything we find out in the clinical material we'll test in the cell lines, and anything we find out in the cell lines we'll test in the clinical material. So we're running a sort of 2 trials, really. Okay, so that's the Ewing story. So the trial's opened. We're trying to recruit 40 patients. It may go to 80 patients depending on the design, and we've started. So that's good. This is the second trial, which I'll skip through quite quickly because I think I'm running out of time. And this is Osteosarcoma. Now here are the Circus plots again, slightly different colors. But you can see Osteosarcoma is much more complex. You can immediately see that. You can see all the lines flying across. And in fact, it is much more complex because there are many, many mistakes. In fact, on average, 2,000 translocations in Osteosarcoma. It's paradoxical because Osteosarcoma is very chemotherapy sensitive, as many of you know. But its genome, the genome within Osteosarcoma is very much disorganized. This presents a number of major problems, one of which is disease treatment resistance. And so the strategies for osteosarcoma are going to have to be different. Any hope that chemotherapy or additions of different chemotherapy over the next 10 years is going to make a difference, that's not going to happen. I think we've reached the ceiling there. Where I think we'll go next is really in trying to understand how our own immune response can start to destroy osteosarcoma, because each of these mistakes presents a new, what we call an antigen or peptide to the body. And what osteosarcoma is doing, it's a way of immunizing. It's a load of cells that are constantly challenging our immune system. And so, yeah.
Sorry, can I ask you a question?
Yeah.
In each patient,
Yeah.
Are there lots of different
Yeah.
gene mutations?
Yeah.
Or is it just that in different patients there are different gene mutations?
In each patient, there are different ones. and they are all different from the next patient. Okay? So trying to find a one-size-fits-all type of therapy is probably going to be very difficult. We can ignore this and just plow on with what we've been doing for the last 20 years, but I think we're making a mistake. So the chromosomes are completely disrupted in the tumors, but what the tumors are doing are setting up a huge inflammatory response. The body responds very positively to osteosarcoma. There's a lot of genes that are changed, a lot of cells that come in, inflammatory cells called white cells in the body that flood into the osteosarcomas. And you can count some of them. These are the macrophages. These are like the dustbin white cells of the body. They go out and chew everything up and destroy it. They're pretty useful. If you don't have them, you're in trouble. But in diagnostic biopsies or resections or metastases, if you count them, there's a huge variation, but they're generally very high levels. And these macrophages can be either M1 or M2, and there's a whole series of mechanisms that can do this, and they're derived from circulating white cells called monocytes in the blood. So the question is, can we activate our own immune system to challenge osteosarcoma and destroy it or regulate it? And there is a way that's already licensed, and it's used as adjuvant treatment. It's called mifepristone or MePACT. It's used after resection of osteosarcoma. And actually, we know how it works. It works by targeting the macrophages, these activated white cells, in a whole series of enzymatic actions to produce a load of hormones. They're given names like IL-8, IL-6, IL-1, TNF-alpha. And so you infuse the mifepristone, it activates the macrophages in the tumors and produces a lot of things that destroy the tumors and activate your immune system to the tumor. So we decided to do a second trial then for osteosarcoma, which is nothing to do with chemotherapy in its first-line sense, but really to try and work out a little bit more what's going on with this MTP, this mifepristone. And so we're doing a trial in metastatic and recurrent osteosarcoma, currently not treated with that drug, not in the license for that drug, to try and find out with the material that we take from the biopsies how the immune system is responding to osteosarcoma. And again, it's the same principle as a Ewing's trial. It's very detailed. It's the same depth of information. There's a little bit more complicated design because of randomization between surgery and no surgery. The endpoints are more or less the same. The effects of the mifepristone we expect, we know about chills, fatigue, headache. These are all represent the effects of these little hormones that are made by the macrophages. That's the outcome. So with that, again, if we can identify what's going on inside the tumors, measure them very carefully, we want to turn this trial into 2 or 3 trials. Again, the same principle. So I'll finish there, and thank you for your attention. I hope I've managed to explain why we're doing the things we want to do. The 2 trials are small, expensive, but have involved major European collaborations. It's what the community feels we ought to be doing as the next stage. And we've got a very small team trying to drive this. At the moment, 5 or 6 people. And we hope we'll be able to come back in the next few years and tell you what we found. Thanks very much for your attention.